Our first presenter, Dr. Alan Gowan, who is an esteemed medical director and chief pathologist at PhenoPath Laboratories, and we're very thankful for him to take his time today to speak to you. Our other presenter, Dr. Farah Patell-Socha, who many of you may have heard a webinar from before, is the senior product manager here at Horizon Discovery, leading the IHC product development. So, without further hesitation, I'd like to pass you over to your very first presenter today, Dr. Alan Gowan.
Thanks very much, and it's a pleasure to be speaking with all of you. I'm going to talk, as was mentioned, about the PD-1, PD-L1 checkpoint inhibitor pathway, which has been a focus of great interest recently in terms of the availability of new drugs which can address this pathway and have had significant effects on certain classes of tumors. So I can click for them. Just trying to see how I advance the slide here. So here we are. Okay, the first slide here shows in a cartoon fashion something about the pathway we're talking about. And the background of this is that we all have a number of immune mechanisms which, at least in theory, can attack and destroy tumors, which often present with neoantigens, so foreign antigens, which induce an immune response. And some of the major players, as shown in this cartoon, are the cytotoxic T cells, which can, with the help of additional immune cells, attack and destroy these foreign antigens and the tumor cells which express them. T cells, as mentioned in this slide, do play a critical role in both anti-tumor as well as antiviral responses. If everything is working appropriately, this immune response can be quite effective in preventing the neoplasms from getting to be dangerous to the host or to the person in which they're growing. However, in some cases, and in some cases for good reason, we want the T cells response to be shut down. And obviously you don't want, particularly in response, for example, to a viral infection, you don't want this response to continue to go on because you can get a destruction of host tissues. And there's a number of normal receptors this T cell function, one of them being the PD-1 ligand PD-L1 pathway. This cartoon from Nature Reviews Immunology last year shows that if you look at this at the cartoon, you can see that the PD-1 PD-L1 pair is actually one of a number of mechanisms which exist on both T cells, which are on the right, and some of the cells with which they interact, in this case given the example of dendritic cells. But as we'll see in a moment, that these ligands by tumor cells. So this PD-1, which stands for the programmed death 1 receptor, which exists on, we've just seen on T cells, on activated T cells. It can be engaged by ligands. We're going to focus on PD-L1. There's also a PD-L2. But these ligands, which can then sort of regulate the behavior of these T cells, can be expressed both by immune cells and in some cases, most critical to our discussion here today, by the tumor cells themselves. And this cartoon expands upon that by showing the T cells, and on the left you can see the PD-1 being expressed by the T cell, but its ligand is expressing here by an antigen-presenting cell, but you can see that there's a whole range of different kinds of cells, both endothelial cells, regulatory T cells, and as we go to the lower right, we can see that they can be expressed by the tumor cells themselves. So it's important to understand that this PD-1, PD-L1 pathway is a negative feedback system that represses the cytotoxic immune response. And under normal conditions, we may want this to occur, but tumor cells have an ability essentially to hijack this mechanism and prevent the T cells, the host T cells, from attacking and destroying the tumor cells. And we can try to address this ability of the tumor cells, in this case the cancer cells. In this next slide, you can see a cartoon by blocking this interaction. And so you see on the left-hand side, you have again the cytotoxic T cells expressing the PD-1 receptor, and the ligand being expressed in the blue dots here on the cancer cell. And that would shut down an immune response if that were to exist. But these new classes of drugs, which can target either PD-1 itself on the cytotoxic T cell or the PD-L1, which is present here on the tumor cell, either one of these can result in aggregation of this negative feedback loop, which would then essentially unmask the T cell to destroy these antigens, in theory. And sure enough, there have been developed and are being developed a whole host of drugs which either address these are often monoclonal antibodies, as you see for the names, or they're sometimes small molecular inhibitors. Either drugs which attack or bind to PD-1, or drugs that bind to PD-L1 have the same effect of aggregating this negative feedback loop. And you can see the two drugs that are in red on the left, nivolumab and pembrolizumab, are two of the drugs which already have received in the United States FDA approval. And we have several drugs on the right-hand side which we expect FDA approval to be imminent. There have been a number of clinical trials going on, and some of the early trials showed that there was significant effect of blocking this PD-1, PD-L1 interaction in a whole host of different tumors. That is not just non-small cell lung carcinomas, but also melanomas, renal cell carcinomas, bladder carcinomas, and even Hodgkin's lymphomas. And more important to the point of our discussion today, many of the earlier studies, and certainly some of the more recent trials as well show, the keynote trials, have suggested that it is this expression of PD-L1 on the surface of the tumor cells which correlates with the response to some of these drugs, and in particular nivolumab and pembrolizumab, which are the two that at this point received FDA clearance. This study back from a number of years ago, which was a sort of a preclinical study, showed that there was, just looking for the safety and activity of some of these anti-PD-1 drugs, that in a series of about 300 patients, as you see in this is, the next one, next slide, we can see the association with response to the drugs, in this case a particular drug, 5H1. You can actually see that the response in patients was only in a subset of those patients who had PD-L1 expression on their tumor cells. That is, there was no objective response seen in patients who didn't have PD-L1 expressed on their tumor cells, as one might have expected theoretically. And actually, looking across a wide range of different tumors, as shown in this slide from a different sort of review paper, you can see that in general, the response rate to the drugs, and this is looking across different drugs, this is actually 4 different drugs in this sort of meta-analysis table. You can see in each case the orange bars showing those tumors which are PD-L1 positive. One saw a significantly improved response to those tumors in those patients who had PD-L1 positive tumors compared to those that did not have PD-L1 positive tumors, which you see in the green bars. So significant effects of the presence of that response. And again, summarizing what makes these drugs particularly interesting is that they are effective across a wide range of different tumor types. So you see melanomas, renal cell carcinomas, Hodgkin lymphomas, and even an interesting subset of colorectal tumors, which turn out to be those which have mismatch repair alterations. So, as mentioned, there are now two drugs, both pembrolizumab and nivolumab, which have received FDA approval, and we expect more to be coming. And we're going to focus on some of the issues relating to pembrolizumab and nivolumab because they have been FDA approved, and they come with specific testing requirements or recommendations, as we'll see. So starting with pembrolizumab, this received FDA approval back in October of 2015 for lung non-small cell lung cancers, particularly those patients who have failed treatment with other chemotherapy indications. So we're talking about a second or third line trial. This is the paper that was published. This is referred to as the KEYNOTE-1 study. And as you see here, this comes from the paper in the New England Journal of Medicine at the same time in which patients were given these drugs. This drug, it was investigated, the tissues were interrogated for the presence of PD-L1, and as you see here on the right hand, particularly on the right hand side at low power at the top and at the bottom right at high power, you can see how this is largely, as one might predict, a membranous antigen which is expressed, which was expressed in a certain percentage of the tumor cells, as we'll see. If you look at this trial, which had, as you see, about over 700 patients, there was a distribution. This is the distribution of the expression of PD-L1 in tumors. That is, about a third or more of the tumors did not express any PD-L1, and there were both low and high expression levels, as you see, defined by the 50% cutoff. So about 28% of these tumors had high levels of expression, whereas about more than a third to 40% had what were defined as low level expressions, between 1 and 49%. And in this study, which was again this KEYNOTE-1 study, what the data apparently showed was that when looking at survival, those patients who had what were called the scores, the PS scores, percentage scores greater than 50%, had a much better outcome than those that either had no expression, that is less than 1%, or expression at the low levels of 1 to 49%. As we'll see in a moment, these data are a little bit different than what was obtained in the KEYNOTE-10 trial, which was more recently published. Now, so based upon these data, when the FDA approved pembrolizumab for non-small cell lung cancer, it approved it along with a companion diagnostic, which was developed in conjunction with the investigators. And as the data showed that greater than 50% or more of tumor cells positivity seemed to be associated with a response to the anti-PD-L1 drug pembrolizumab, the FDA approved the companion diagnostic, which was an immunohistochemical test, which was produced by DACO, called PharmDx 22C3, 22C3 being the name of the clone designation. So presumably for use of the drug, if you look at the package insert, we'll see in a moment, it required a greater than 50% or more of the tumor cells to be positive. Now here's the more recently published KEYNOTE-10 trial, which showed a little bit different results, again showing that there certainly was a response. And this looks a bit different, two different doses of pembrolizumab compared to docetaxel. And again, if you look at the left-hand side, patients who were treated with a pembro had a significantly better survival outcome at these cutoff dates. You see here, but this was actually also seen in patients who had lower levels of PD-L1 expression, so that there was a response seen, as you see on the right-hand side, and only patients who had positive PD-L1 positive tumors were included in this trial, so that there are no patients here who had less than 1% or no expression of PD-L1. So these include both low and high level expressors, and unlike the KEYNOTE-1 trial, we do have a response even in the lower level expressors of PD-L1, which is important to note. That's not to say that there wasn't a difference. If you look at this from the trial, you can see, I think the next slide shows that there was a more hazard ratio was certainly higher in those patients who had greater than 50% of their tumor cells positive, but there was a lower but still clinically significant response in those patients who had lower level expressors. So, as I mentioned, there were no PD-L1 negative patients included in the KEYNOTE-10 trial, but all patients benefited, including those showing both higher and lower levels of survival. Right now, the scoring is based again on the initial KEYNOTE-1 data, which requires either requires membranous staining, as I showed you in that earlier slide, but only tumor cells showing membranous and not cytoplasmic signal is counted, and the level of intensity of the immunostaining in this scheme is not included. And here's from the package insert, again based on the KEYNOTE-1 trial, you see that there are 3 levels of tumor expression which are defined, no expression, which is defined as a score of less than 1%, a low level of expressors, which are those between 1 and 49%, and those so-called high expressors. And in the initial configuration of this, the way this is, has been recommended to be reported, patients that have higher levels of expression, 50% or more, those considered candidates for Keytruda, which is pembrolizumab. However, based on the KEYNOTE-10 trial, many oncologists want to know if there's lower level expression as well, that is the 1 to 49% group. This is sort of an evolving, I think it's an evolving scoring system, which will probably be modified as more data are received. I have some images now to show you. This is a non-small cell lung carcinoma. There's an H&E, and in this next slide you can see the what a strong positive looks like. It will advance. There it is. You can see this strong membranous staining in this case in the vast majority of the tumor cell population. This is considered a high positive or greater than 50% positive lung tumor, non-small cell lung tumor. Here's another tumor which has actually some a considerable amount of necrosis, particularly on the upper right and lower left. Is the H&E. Again, though, in the viable tumor cells, the vast majority of the tumor cells show strong membranous PD-L1 expression. So this is, again, another high positive PD-L1 tumor. Some of these tumors get a bit more difficult to evaluate, as you see here, with the arrow is shown in the lower, in the sort of center, lower center. You can actually see that there is tumor necrosis. And when you look for PD-L1 expression in this case, there are certainly clusters of cells which are positive. There's areas in the upper right here which probably correspond to necrotic tumor cells, and we don't score the PD-L1 expression there. We have to limit our sort of the denominator of looking for PD-L1 positive cells amongst the viable tumor cells. And here, this would present a great challenge to know whether there is in fact 50% of these cells positive, or if it corresponds to the lower range of 1 to 49%. And it's, as we discussed earlier, since PD-L1 can be expressed in many cells of the immune system themselves, for example, macrophages or antigen-presenting cells, it's important that when you look at the denominator that you're actually counting just tumor cells in there. The requirement for the FDA-approved assay is that one counts at least 100 viable tumor cells. And if there are less than 100 viable tumor cells, that you really can't give a valid interpretation. And that can be a problem in some cases. Here's another example of a tumor in which we were sent to evaluate. This is in a pleural fluid cell block, and we do have clusters of what at least one might identify as positive tumor cells. However, as you know, in the context of pleural fluid, we have a lot of inflammatory cells. We can have reactive mesothelial cells. So it's important, again, to know which cells one's looking at. Here's what it's just a higher power H&E, in which there are cells which could represent clusters of tumor cells. They also could represent, some of them could represent reactive mesothelial cells. And when we looked at the PD-L1 in this particular case, there are certainly strongly positive cells scattered throughout here. There are clusters of cells which do not appear to be positive, such as here. I think I have this at a slightly higher power over here. But remember that the scoring system doesn't actually have us look at the intensity of scoring. So even weak positive cells, such as the ones you see here, which could be considered positive. But the question is, are these tumor cells, or are these mesothelial cells, or are these macrophages? Here it is again at high power. And so in this particular case, we would investigate these for looking for expression of markers which would be help us identify just the tumor cell population. And you see here TTF1 being a good marker for the nuclei of tumor cells. So this confirms that these clusters, at least many of the clusters, are in fact tumor cells, although some of the others, which we might have suspected were tumor cells, may not be. And here again is this shows TTF1 expression that we have isolated tumor cells, but most of the cells in this field do not appear to represent tumor cells. In fact, by using a marker for another cell type, in this case CD163, a macrophage marker, we see that the vast majority of cells here are actually macrophages. So this shows you some of the challenges that we have in terms of identifying the tumor cell population in certain contexts. We also have a problem that often the PD-L1 testing is done at the very end of a cycle where the patient's tumor has already been tested for EGFR mutations or ALK alterations, and so there can be very, very little tumor cell left. And so it's even more important that someone who's looking at this and scoring the PD-L1 tests knows exactly what cells are being examined and where this positivity is. Turning to the other drug that's been FDA approved, nivolumab, it has the same target, which is PD-1, and has been demonstrated. The drug was approved back in 2015 for squamous non-small cell lung cancers, and was extended to all non-small cell lung cancers in October of last year. Again, this is a second and third line drug at the current time. And in the study that was published, subgroup analysis indicated that the level of PD-L1 expression in the tumor may in fact help identify those patients with longer survival. And so again, in approving the drug nivolumab in the context of non-small cell lung cancer, the FDA simultaneously approved, in this case, a different assay, also manufactured by Dako, but uses a different monoclonal antibody, in this case 28/8 instead of the 22C3. There's the paper that was published back in 2015 showing the efficacy of the drug. This paper showed that, again, there was benefit to the nivolumab compared with other drugs such as docetaxel, and this actually shows data from the study. On the top in the blue bars, you can actually see the response of patients, again, with being treated with nivolumab compared to docetaxel on the bottom in the green. And you can see that all the individual patients who have black arrows at the end of it are patients who were still responding at the end of the study. So there was a remarkable response compared to docetaxel. And you can see that there were even survival data at 1 year showing significantly improved survival. And the survival benefit was seen in patients regardless of their expression levels. Now, this is important because in this particular case, a different scoring system was employed by the investigators compared to the 22C3, in which we had that 50% cutoff. Here, there were expression levels were recorded as either being 1% or higher, 5% or higher, or 10% or higher, and that got incorporated into the scoring system that was recommended for this diagnostic test, in which the pathologist is required to look at the PD-L1 expression as defined by the percentage of cells. So a negative result is essentially in the top line, which is less than 1%. And then we score them as whether they're representing greater than 1%, or greater than or equal to 5%, or greater than or equal to 10%. It's important to note that all these three categories in the bottom are considered positive, but they're still recorded that way. Now, when the FDA approved this, it did not approve it. It approved the test as what's called a companion diagnostic, as was the 22C3 assay for pembrolizumab. This was a category that the FDA defined as something called a complementary diagnostic, which, as noted in the DACO scoring sheet, that the complementary diagnostic may provide additional information, but it is not required, as a companion diagnostic is required for the, as with 22C3 and pembrolizumab, is required for the treatment with the drug. In this case, a complementary diagnostic means that it's useful additional information, but it's actually not a requirement for the prescription of the drug. And this just summarizes some of the data in terms of the different drugs, some of which are in the pipeline, some of which have already been FDA cleared, like pembrolizumab and nivolumab, the first two columns. Each one of these drugs, the two you see here, the atezolizumab and the durvalumab, are expected to be FDA approved soon. But as noted here, there's going to be a different companion diagnostic, which we think, which will be on a different platform, using a different antibody, using different scoring systems. So we expect, even though things are a little bit strange and complicated with pembrolizumab and nivolumab, at least we expect things to get a little bit even more complicated as these other drugs come through the pipeline and get FDA approval. So we anticipate that there will be even additional antibodies that are used for the test, and different cutoffs, and complicating everybody's life even more. But right now, we can focus just on pembrolizumab and nivolumab, the two drugs which have FDA approval. And it's going to present a particular problem to our clinical colleagues, knowing, and pathologist colleagues, knowing which assay to perform, because there are, as we know now, there are at least two different assays, the 22C3 and the 28-8, and these are required only in the context of certain cancers for which the FDA has approved both the drug and in the testing system. So you see here in this flowchart that we've put together, the first question is whether something is non-small cell lung cancer, and that will trigger, if the patient's being considered for pembrolizumab, the 22C3 assay. But if the patient's being considered for nivolumab, then there is the 28 assay, which, as we mentioned earlier, is a complementary, not a companion diagnostic. More recently, the FDA extended approval of nivolumab to melanoma, in which case, again, it has would go along with the 28-8 complementary diagnostic. But in other tumors right now, there is no requirement for, at least at the current time, for companion or complementary diagnostics, and we have there are the assays that we call more generic assays, which can also be employed. So it's a bit of a complicated business, which is expected, unfortunately, to get more complicated, but that's just, I guess, the nature of the beast for this particular type of testing and the use of PD-1 and PD-L1 inhibitors. So I will stop here and thank everyone for their participation and for their time, and be happy to entertain questions.
Thank you, Joe, and thank you, Alan, for this excellent introduction and insight into the world of PD-L1 and PD-1. I'm Farah Patell-Socha, and I'm Senior Product Manager leading the IST development program at Horizon Discovery, which is a company based in the UK in Cambridge. For the next 15 minutes, I will be talking about measuring and improving the reproducibility of your PD-L1 IST assay's technical performance. As you all know, immunohistochemistry has evolved from an investigative tool into a widely used diagnostic technique with clinical laboratories. IST serves as a diagnostic and prognostic assay, and in a clinical setting, it increasingly contributes to the choice of treatment. Although it has been employed for over 50 years, the application of this method suffers from a lack of standardization and reproducibility among different laboratories. So today I'm going to touch upon the variability arising in a typical IST workflow and how we have developed these genetically defined IST reference standards as independent quality materials for everyday use to address the issues around consistency and reproducibility of your assays. I would like to highlight at this point that these standards are for research use only. I will also present a couple of short case studies on PD-L1 and how defined standards can be used to identify assay failure in your lab. So a little bit about Horizon before we get into PD-L1 and the reference standards. Horizon Discovery is a cell building company. We are a company that looks at all aspects of cell function. We drive to understand the interplay between the DNA, RNA, and protein, and how these aspects impact the laboratory workflow, be it an RNA, NGS, DNA, protein. It can be any workflow. Once you understand the cell and its function, you can apply it to anything. Horizon builds human disease models and reagents derived from these genetically engineered cells that our customers use, or we can also deploy them on their behalf to gain knowledge of the genetic drivers of disease, develop new novel drugs or cell therapies targeted to these genetic drivers. And most importantly, from our side, and what we're going to address today, is we are extremely passionate about developing and delivering high-quality, genetically defined standards for users' independent external control. So from this understanding and using our gene editing technology, we have developed a range of reference standards for DNA, as you can see in this on the slide on the right-hand side, RNA, fish, cell-free DNA, and IST workflows. Today, I will focus on the IST reference standards, their development, and their utility in the IST workflow. And I will also go into a little bit of detail in a couple of slides on how we've developed the PD-L1 reference standard. So, for some of you who have already heard me talk before, I know I'm repeating myself here again, but I believe that this is a very important question, and before I begin my introduction, I would like to ask you to keep this following question in your mind. And most importantly, when you run your IST assay in the laboratory, be it in a hospital, a reference lab, an assay development team, a CRO, so what is the impact of assay failure in your lab, and how do you monitor for it? It is an important question to ask when you are routinely running these assays, and it becomes even more critical when you're running markers like ALK4, HER2, and now most importantly PD-L1 laboratory. What would happen if your assay gave you a positive or a false negative, and what steps would you take to mitigate against that? So this is a very standard basic scheme of a workflow. And what I want to highlight here is where can variability creep in. And some of the people we work with, surprisingly, do not keep not understand this workflow, and and where the variability can creep in. So it starts basically with your tumor sample and your fixation method and time. You have your antigen retrieval, which is also a very important step, your antibody staining and your choice of antibody, the amplification and microscopy, and more now and more you see the quantitative digital pathology aspect, where you quantitate the intensity of staining per cell and you analyze that, and then an action is taken. I won't particularly go into details as our previous Horizon CAP webinar, which was given by Professor Clive Taylor and David Grimm, went into detail on the variabilities of these workflows and to each parameter in detail. So the important point is understanding each step, be it the fixation, the choice of antibody, the choice of the assay, is important, and understanding this will help you understand where the variabilities can creep in. So, and this brings us to the challenges facing the clinical pipeline, including PD-L1. The importance of having a reliable, consistent, and accurate assay is crucial at all stages. You have the research and development stage, which is right at the beginning. You have your clinical studies, all your clinical trials, patient stratification, which is also used, basically taken care of. I mean, what I'm trying to say is the assays impact patient stratification. And then finally, you have the market. You have your proficiency schemes, you have your companion diagnostic kits, and the ring trials that help with harmonization and standardization of these assays. So good assays validation leads to a better trial design and improves chances of patient selection, market success, and most importantly, delivering the right diagnosis and prognosis to the patient. Unnecessarily can at best lead to more time and cost to get something through, and at worst can result in the program failing. Bringing in more rigor, we believe, in the clinical development phase and bringing in standardization and harmonization early on, very early on, is necessary. So our objective in Horizon was to try and answer some of these questions by developing these precisely defined reference controls with negative and positive cell lines on the same slide. So the idea what we wanted to do is to have a system where we had the cell line controls, you had your negative, low, medium, high on the same slide, and you could put your sample of choice on your test sample on the same slide and use it as an on-slide control for every single test that you run during your optimization and validation step. So and by doing this, you will start to probably gain an understanding and answer some of these questions like, what is the sensitivity and specificity of my antibody? What can I use to set a threshold if I was using image analysis? Is my validated protocol optimized? Is my test sample a false positive or a false negative? The fundamental question basically you're asking yourself by using a control is, has my IC assay worked at that particular time? And again, as I said in the past, I would like to highlight that these reference standards are for research use only. So what we did was, leveraging on our gene editing technology and understanding the variability in workflows, we developed standards that had these highly characterized negative, low, medium, high protein expressing cell lines. And with that, one could assess the technical performance of your assay with these known protein expression levels. And most importantly now, with PD-L1, one could establish the practical lower limit of detection of your IFP methodology with validated low protein expressing cell lines. And I'll give you a couple of examples of that in the next few slides. And most importantly, guide the evaluation of antibodies based on their sensitivity and specificity. So the other thing we did recently, we work with the company called Visiopharm, and they are quantitative digital pathology providers. We use their system and their software to further QC these cell lines. Biomarker-specific algorithms are built that allow us to characterize and assess each of these negative, intermediate, and strong protein-expressing cell lines in our standards. In the next couple of slides, I will discuss the development of the PD-L1 standard and demonstrate their role in highlighting variability in the workflow. Yeah. So Alan has gone through this slide in detail, but my intention here was to give you, it's complex, as a reference standard provider, we've been getting a lot of questions, especially from laboratories who are trying to make sense of these assays, what drugs should they use, but most importantly, what assays should they set up in their lab. So at the moment, most laboratories are performing LDTs. They are comparing the different assays internally, and they are struggling. So, immunotherapy has become an increasingly appealing therapeutic strategy for patients with cancer, with many late-stage clinical trials, as we know, in different cancers, melanoma, prostate cancer, lung cancer, but most recently, in NSCLC has become a focus for the next generation of human-based therapeutic strategies. Currently, there are multiple PD-L1 drugs in the same class, its own companion diagnostic, as Alan described in his presentation. The problem is not so much a regulatory problem, but a downstream challenge for pathologists to forming the test for different assays. Like I said before, community oncologists who have to figure out the best test to use amid time constraints with limited understanding of genomics and often with insufficient data comparing platforms. And in order to determine if the patients will respond to specific treatment. And these ideas basically led us to develop a new technology at Horizon, which had actually one of the things that has never been done until today, is creating a standard that has controlled protein expression levels. And that would help, and our idea was that would help us to answer some of the questions around standardization, harmonization, by developing for the first time PD-L1 represented with these negative, low, medium, strong, controlled protein expression expressing cell lines, which would test basically the sensitivity and specificity of the assays. Do they all behave the same, and what are the differences? I won't go into much detail, but the next slide here kind of shows you what technique and what was this landing pad approach that we used. So a platform was developed where we used promoters of different strengths, which are these arrows, purple, blue, pink, dark purple, and gray, on the extreme left-hand side. I don't know if you can see my arrow. 1 second, please. Yeah. So we had these different promoters here with different strengths driving PD-L1 expression. You had, and you also had a cell line with PD-L1, without PD-L1 expression. So you had the parental cell line, which would act as the negative control, and then you had all these different 5 other cell lines. And from this, we generated many clones, engineered clones, which went through a series of molecular and protein characterization before the final selection of cell lines were made and fixed, and we embedded them into FFPE using Horizon's proprietary methodology. And finally, we got this block. We cut a 3-micron section, put it on a slide, and we selected about 15 cell lines here from these codes, and we hoped that they had the negative, low, medium, high expression. We also assessed the cell lines using QDT, and I will get to that in a bit. So the idea here was, again, to have a range of the negative, the 1+, 2+, 3+, is basically a low, medium, and a high expressor. One second, please. Yeah. So we got very excited when we saw our first IHC results using this technology. So like I said before, we had these 15 and we had selected these 15 engineered cell lines, and we were hoping that we would have the high expressors that we got. So we had 3 clones here that expressed extremely high amount of PD-L1 protein. Then you had this beautiful intermediate range where you had the lows and the intermediates. As you can see here, this is a very nice intermediate. You had wonderful clear membrane staining around each cell. And then you had finally the parental cell line, which was your isogenic matched parental, which does not have PD-L1 expression. So this is a very important cell line because this has no PD-L1 expression, and this has PD-L1 expression. So this gives us a very good start. So what I also wanted to add here is we at Horizon, we used the cell signaling antibody, which was, and our scientists had developed an assay for that antibody. The 3 micron sections were cut. We used the anti-PD-L1 E1L3N at 1:200 dilution, and we ran it on the Lycobond immunostainer, and you can see these pictures here. Now, the next stage was, do we see the same expression when we run another antibody? So we took SP142 from Spring Bio, and we again developed our own assay internally. So you have the same wonderful PD-L1 membrane expression across the 15-core array, except for the negative cell line here. But these are also engineered negatives. And so it matched the the cell signaling results very clearly. You had the highs, you had the same intermediate low expressors, and then you had the negative cell line. This gave us a lot of confidence that we were on the right track. And what we do at this stage of development, we always send this out to multiple laboratories, and assay developers and key opinion leaders to test these assays using their protocols. And I will come to that in a bit. So we were happy, as I said, the results were highly concordant, and both arrays were sent to a number of key labs, and all 4 assays that are currently available, the approved assays, were tested. So, as you can imagine, building reference standards is a big responsibility for us, and hence we have stringent QC criteria at Horizon. We adopted image analysis to further define the positive and negative cell line cores, and that has allowed us to monitor our batch-to-batch reproducibility studies and the assays routinely. In this case, the intensity of individual cells in the cores were assessed using VisioPharm software, and the resulting histogram were compared to create the optimum cutoff between positive and negative cells, minimizing false negative and false positive results. The thresholds were then set and used for the ongoing analysis to ensure consistency and reproducibility. So the 8th score provides additional information about the staining, and it is a score of the intensity, staining intensity, and can monitor changes more accurately than a simple positive and negative cutoff. But we also have the percentage positively for each of these cell line cores. Now, as you can see here, is just give me a second. You have a wonderful dynamic range. So the range that you saw in the previous slide, where you had the high top 3, which were your high expressors, have a higher 8 score, and then the negative is your negative, and then you have this wonderful range in between. So our engineering worked wonderfully, and we had the negative, low, medium, and high expressors that we were looking for. Now, we often get asked and about when we talk about cell lines and is the batch-to-batch reproducibly. What if I were to regrow a cell line? Would you get the same results? Can you ensure reproducibility? Now, we always run reproducibility studies as part of our process. And what we decided to do is we took those 15 cell lines, we selected 11, and we regrew them. We remanufactured the raw blocks. It went through the entire system for a second time. And the cell lines from the original cell line screen were selected, grown, fixed, manufactured. 3 micron sections were cut and tested internally and externally. And as you can see in this image, every single cell line core behaved exactly the same. You had your 3 top high expressors. Now the only difference between this 11 core array and the 15 core is just that we've taken down 3 cell lines, and we reduced the number of cell lines because our ultimate aim was to select a negative, a low, a medium high that I have highlighted in this in this slide as in blue. And as you can see, a wonderful dynamic range is again observed in these cell lines. So this was your 15-core, and that was your 11-core, and this again shows that it is absolutely the same. But what we wanted to do, an IHC assay just isn't enough. We wanted to do QDP on them and actually compare from batch 1 and batch 2. So what we did next was to combine the 8 scores from individual runs from batch 1 and batch 2 for negative, low, medium, and high protein expressing cell lines that were highlighted in blue in the previous slide. And as expected, both reproducibility batches were almost identical. But here you can see is you have batch 1 and batch 2 are exactly the same, and very little difference between them. And the error bars, most people think that these error bars are to do with cell growth, but actually this is a variation that you see when we run our assays. Actually the end number for this was 10. So, and which makes sense. The top that are your high expressors have very little variability, and this is very important out in the low and the medium, and this is what will test the sensitivity of your assay. So, 4-core PD-L1 standard was manufactured now, containing those 4 cell lines that we know work very well in batch 1 and batch 2. And then we define the 8 scores. We define the percentage positivity in each of these cell lines. And again, they were tested internally and externally, and the QDP shows batch to batch reproducibility and consistency of these standards. Now, as I mentioned earlier, we work with external key opinion leaders, reference labs, who are experts in IHC to externally validate our reference standards. And most importantly, test the different platforms that are out there. As you know, platform also can lead to a different result in staining intensity. So we, through our alpha and beta development programs, we performed many studies, and this is a very interesting exercise for PD-L1. The standards were tested using all available antibodies and assays currently in the market. One of the first studies we did was with PhenoPath laboratories in collaboration with Dr. Vegan Fulton from PhenoPath. And they used our reference standards amongst their own tissue controls to optimize and validate their assay. As you can see in the slide, you have our 15-core array here from Horizon, which is the development array containing all these engineered clones. And they added their own tissue control sample, a PD-L1 positive sample on the slide as an on-site control to add confidence in the assay. And you can see the very nice dynamic range that is observed. But in the next slide, I highlight that we see exactly the same that we saw using internally at Horizon. You had the high expressors, you had the dynamic range here, you had intermediate, and you had a very clear negative. And this matched the tissue staining as well. So if, for example, if this negative cell line here was positive, then you know immediately that something went wrong in your assay, and this is a false positive. So this is beautiful, this is very clear, and the assay had worked wonderfully. Another case study we have is a study with an external laboratory. And in this case here, you have, and this highlights the assessing the sensitivity of the assay. Here, this is our Horizons result. You have the high, medium, low, and negative. Very clear membrane staining seen in all these different cell lines except for the negative. And then you have the external laboratory staining where they lose sensitivity using the same antibody that we did, but using a different protocol. You have loss of sensitivity. So they do not pick up any of the low and medium expressors. So if you were to go back, for example, to this slide, and if they had done that, what they had was they had the positives, the strong positives. They picked up the negative, but everything in between was negative as well. So this was very interesting, and we found these results coming in from many labs who not only use this antibody, but whose assays are not optimized enough. This is another case study here. This is using This is using the 28A antibody. This is again, the one thing I do want to add here, these are all LDTs. The external laboratories who have the examples that I've shown here are, in these two cases, are LDTs. So that these are assays they are performing themselves, are trying to optimize at the moment. So here you can see the negative has background, it has also memory staining, and this clearly shows that the assay is not optimized. So this particular laboratory was trying out 4 different assays internally, and when they sent us their results, all 4 assays looked different on the same reference standard. So that again shows that having a consistent, reproducible standard alongside when you're optimizing and validating your protocol is crucial. So to summarize, what we've done now is we have created these PD-L1 sections and slides as a tool to optimize and validate, but most importantly for laboratories and hospitals running PD-L1 routinely, we have put them in we actually provide FFPE blocks that are cost effective most importantly, but also they provide a section into reporting control. So you can cut the minutes just before you go to mount your slide and use it as an on-slide control during your optimization and validation process. So as I mentioned earlier, you have the defined cell line controls. These are extensively engineered and characterized. They offer an inexhaustible supply of consistent resource material, and you test your analytical performance. It's important for methodology transfer, and it helps you to establish the practical lower limit of detection of your IIT methodology. To end, the final question I would like to ask you is, what is the impact of these failures? Are you facing these problems internally at the moment, and how are you testing the technical performance of your assay, and how are you testing the limit of detection of your workflow? Thank you.